Hello, and thanks for stopping by to check out my poster. In the next few minutes, I'm going to talk to you about the assay we developed to track several human memory CD4 T cell subsets within antigen-driven responses. Activation-induced marker, or AIM assays, are a commonly used tool to detect antigen-specific T cells. But the very nature of activating the cells is changing the T cell phenotype, and not only for these markers of interest to define the antigen-specific cells, but also for key lineage-defining markers. This is a particular problem for regulatory T cells, which are usually defined by their high expression of CD25 and low expression of CD127. They also express the transcription factor FOXP3, and they can be distinguished here from their non-Treg or T conventional cell counterparts. However, upon activation, conventional T cells express CD25, transiently express FOXP3, and downregulate CD127, so post-activation we cannot use these markers to accurately define Tregs. Our solution was to sort cells prior to any activation occurring and label with cell proliferation dyes. So we sorted the memory T conventionals and labelled with cell trace violet, and the memory Tregs into the CD39 negative compartment and labelled with CFSE, and the CD39 positive compartment and labelled with cell proliferation dye EFLOR 670. We rested these overnight separately and then reconstituted together with autologous antigen presenting cells. And we can see that the same ex vivo ratios here were maintained when we gate on total CD4 T cells. We then performed our AIM assay, which after 2 days incubation with antigen, the upregulation of CD25 and OX40 allows the antigen-specific cells, in this case in response to a CMV peptide stimulus, to be accurately gated and analysed. We can now track these 3 labelled populations within our antigen-specific response. And you can see that even though the T conventionals are the dominant cell population within the CD4 T cells in the well, when we gate on our antigen-specific cells, as we had previously published, for these antigens, it's actually the CD39 Tregs that dominate the response. We can then let the assay go to day 6 and analyse cell proliferation. And here we see that both Treg subsets had negligible cell proliferation. Instead, the only cell proliferation was occurring within the T conventional cell subsets, and this was true for a variety of cytomegalovirus and herpes simplex virus antigens, as seen over here. So in summary, this method enables the parallel analysis of multiple CD4 T cell subsets within antigen-specific responses. We can accurately analyse both Tregs and T conventional cells, and we can do this within early activation and proliferative responses. This will enable more precise studies of human memory generated following infection or vaccination. Thank you.